Again, later we shall see the role or the importance of anaerobic respiration. This now brings us in detail to the process of respiration. We have seen so far the importance of respiration. We have seen the two types of respiration. We have seen the products of each type of respiration. We now move on and look at the process of respiration. Like we said, the main respiratory substrate is glucose. But remember the beginning we saw that lipids, proteins, carbohydrates can all be broken down in the body cells to release energy. How does this occur? That is what we want to see in the process of respiration. However, we are aware that animal cells store carbohydrates as glycogen. Plant cells store carbohydrates as starch. If respiration is to occur in animal and plant cells, the stored starch, the glycogen are all converted to glucose. They are all broken down to glucose. That is why we are saying that the glucose, which is the main respiratory substrate, in the process of respiration, is broken down in three main stages. Therefore, respiration occurs in three main stages. The first one, called glycolysis. The second one, called the Krebs cycle, also known as the tricarboxylic acid cycle. The third stage is the electron transport system. Glycolysis is the first stage, and it occurs within the cytoplasm of the cell. It takes place within the cytoplasm. Krebs cycle, or the tricarboxylic acid cycle, occurs in the matrix of the mitochondrion. You remember that cytology. While the electron transport occurs on the stocked particles of the inner mitochondrial membrane. These stocked particles are what we refer to as the oxysomes. The oxysomes. Now, talking about these parts where the different stages occur, we have seen that glycolysis occurs in the cytoplasm. Krebs cycle occurs in the matrix of the mitochondrion. This structure here is what we are calling the mitochondrion. A cell has a variety of mitochondria. The mitochondria have inner membranes and outer membranes, but the inner membranes fold into what we call the cristae, with another part known as the matrix. So that is the matrix of the mitochondria. But glycolysis occurs within the cytoplasm of the whole cell. Now, when we look at this cell, remember our cytology. A cell has a variety of parts, just like we can see. But now, because we are focusing on respiration, our focus is mainly on the mitochondria together with the cytoplasm, because it is the cytoplasm where glycolysis occurs. Krebs cycle occurs in the matrix of the mitochondria, while the electron transport system, we shall see the detailed structure of the mitochondrion later, where we have the stocked granules on the cristae where electron transport takes place. So let us get familiar with the sites where they occur as we move into the details now of the processes or of the stages of the process of respiration. So we move to our first stage, and that is glycolysis. Therefore, if we are asked to describe the process of respiration, we need to note that it occurs in 3 stages: glycolysis, Krebs cycle, and the electron transport system. We now move on and we begin by looking at glycolysis. Now, the whole glycolysis can be summarized very well in that flow chart or in that cycle. So we can use this to understand clearly how the process of glycolysis takes place. However, glycolysis, we have the sugar, glucose, being broken down into 2 molecules of pyruvate. Glucose is a 6-carbon compound, while pyruvate is a 3-carbon compound. Therefore, we define glycolysis as a series of reactions in which a glucose molecule is broken down into 2 molecules of pyruvate or pyruvic acid. A series of reactions in which a glucose molecule is broken down into 2 molecules of pyruvate. Why are we saying so? We can see that we are beginning with glucose as a main respiratory substrate, but in the end, we shall end up forming 2 pyruvate molecules. Therefore, remember I told you, starch is the storage form of carbohydrates in plants, glycogen in animals. They are all converted to glucose. Therefore, glycolysis begins by glucose getting phosphorylated. A glucose molecule is phosphorylated, i.e., or that is to say, addition of a phosphate molecule from ATP, adenosine triphosphate, liberating adenosine diphosphate, forming glucose 6-phosphate. This occurs in presence of an enzyme called the kinase enzyme. Glucose is phosphorylated. That is, a phosphate group from ATP is added to glucose, forming or liberating adenosine diphosphate and glucose 6-phosphate in presence of kinase enzyme. Meaning when a phosphate adds to glucose, we simply form glucose 6-phosphate. Glucose 6-phosphate then undergoes isomerization, or what we call isomerism. Isomerism means reorganization of the atoms within the glucose 6-phosphate molecule. In other words, atoms get reorganized, a process we are calling isomerism or isomerization. This occurs in presence of an enzyme called the isomerase enzyme, this time forming fructose 6-phosphate. Glucose is phosphorylated by addition of a phosphate molecule from ATP, forming ADP and glucose 6-phosphate. Glucose 6-phosphate undergoes isomerization or isomerism, forming fructose 6-phosphate, which is further phosphorylated. An additional phosphate molecule is added. The phosphate is from another ATP molecule, forming or liberating ADP and fructose 1,6-bisphosphate. Why are we calling it 1,6? Remember, the other one was fructose 6, meaning the phosphate was at carbon atom number 6. But now when another phosphate molecule is added, we now form fructose 1,6-bisphosphate, meaning that we are now having two phosphates, one in carbon atom 1, another one in carbon atom number 6. Hence, at that point, you have fructose 1,6-bisphosphate. However, fructose 1,6-bisphosphate is unstable. It is unstable. Therefore, it splits in presence of an enzyme called aldolase enzyme. It splits into two molecules of glycerate 3-phosphate. Glycerate 3-phosphate is also called triose phosphate. Glycerate 3-phosphate is also called triose phosphate, which is a 3-carbon compound. Glucose is 6-carbon, glucose 6-phosphate 6-carbon, fructose 6-phosphate 6-carbon, fructose 1,6-bisphosphate 6-carbon. It splits into two molecules of glycerate 3-phosphate or triose phosphate, whereby each is a 3-carbon compound. Remember, we are looking at the process of glycolysis. We are moving step by step. The glycerate 3-phosphate, also known as the triose phosphate, undergoes additional phosphorylation. This time, an inorganic phosphate is added. At the same time, it also undergoes dehydrogenation. Dehydrogenation, hydrogen atoms are removed. Dehydrogenation occurs in presence of an enzyme called dehydrogenase enzyme, dehydrogenase. And when hydrogen atoms are removed from glycerate 3-phosphate, they are accepted, they are taken up by NAD, nicotinamide adenine dinucleotide. When nicotinamide adenine dinucleotide takes up the hydrogen atoms from glycerate 3-phosphate, NAD now becomes reduced, forming reduced NAD, what we give as NADH2. Glycerate 3-phosphate is phosphorylated. An inorganic phosphate is added. At the same time, it undergoes dehydrogenation, whereby hydrogen atoms are removed from each glycerate 3-phosphate, from each triose phosphate. The hydrogen atoms removed are taken up by NAD, nicotinamide adenine dinucleotide, forming NADH2 or reduced NAD. Remember, we have two glycerate 3-phosphate molecules. So what is happening to one glycerate 3-phosphate molecule is also happening to the other. When glycerate 3-phosphate undergoes dehydrogenation and phosphorylation, we form glycerate 1,3-bisphosphate, also known as triose bisphosphate. Glycerate 1,3-bisphosphate, also known as triose bisphosphate. Now the triose bisphosphate or glycerate 1,3-bisphosphate loses a phosphate molecule. In other words, it undergoes dephosphorylation. When a phosphate molecule is removed, the phosphate molecule is taken up by adenosine diphosphate, liberating the first ATP molecule directly. Glycerate 1,3-bisphosphate or triose bisphosphate loses a phosphate group. That phosphate group is taken up by, it combines with ADP, adenosine diphosphate, liberating the first ATP molecule directly, also forming glycerate 3-phosphate. Glycerate 3-phosphate also loses another phosphate molecule, which is also taken up by ADP, forming